Okay, good afternoon, everyone. Thank you so much for joining us today for our COVID Q&A with Dr. Gupta. Dr. Sanhita Gupta is an associate professor of biological sciences here at Kent State Geauga in the Twinsburg Academic Center. Dr. Gupta earned her doctorate in molecular virology from Case Western Reserve University. Her thesis work involved studies on human parainfluenza virus. Previous to Kent State University, she worked at Case Western University and at the State of Louisiana Public Health Lab as a research scientist. In her classes, she enjoys interacting with the students and assisting with practical hands-on activities in the laboratory. Today, Dr. Gupta will talk to us about the COVID-19 virus, its variants, how to stay safe, and how vaccines work. If you have any questions about the scientific perspectives of COVID and its impact on our everyday lives, please submit them in the chat. I'm going to be monitoring them, and then I will be reading them to her throughout the presentation. So please join me in welcoming Sanhita. We're so glad you're here with us today. Thank you.
Thank you, Dr. That's very good. Special thanks to Dr. Scotsbury because this event would not have happened without her help. And thanks also to Kevin and Amy and all of you who are here today. And we know that pandemic has taken a very important part of our lives. We have been under this for almost 2 years now. And, we thought the end was near, but it's getting farther away as we are progressing. So, and also everything is work in progress. So information is changing as we go. So I'm presenting what I know right now, and there's so much information out there. So it is hard for people I know to figure out which one is the right information and which one is not the right information, because everybody has an opinion on this, and the press is not helping in some ways. So I think we should listen to the scientists because they have evidence for it, and we should only listen to the information that makes sense to us. So all of you are educated, and so you can actually make the difference between misinformation and good information or correct information. But there are people out there who do not know the difference. So I guess it's part of our responsibility to the country and to the world to also tell them which is right or which is wrong, or convince them rather. And so all of us have part in this. This is not working.
Coming to the rescue.
Sorry, I think that's going to work. So in my first slide over here, I have the COVID virus. So we all know this is coronavirus 2, actually, SARS coronavirus 2. We call this coronavirus 2 because we had another SARS long time ago, 2003, I believe. So this is the second one that resembles closely that other virus, the same family of viruses. We are also, I want you to focus on this image, not only on this image, but on the red spiky things that are coming out of the virus. This is why it's called the coronavirus. It's like the surrounding halo that is formed by the spike under the electron microscope. That is how it looks. And these spike proteins are very important, and we're going to talk a little bit more about this as we go on. So, I want to draw your attention to the spike proteins. These are called spike proteins, and they protrude out of the virus. And this gray thing that you're noticing over here, and you've seen this many times, this is actually made out of fat. Okay, so this is a fat layer, and these are proteins that are sticking out of that fat layer. And inside, what is inside? Okay, this is the coronavirus structure. So if you go inside, take a cross section. So you see the spike proteins here in teal, they are marked S. That is very, as I said, very important. I'll tell you why. And then we have the genome inside. So you look at the virus, it doesn't have any machinery to operate on its own. So it can do nothing when it's outside. So if it's sitting out here on this tabletop, it cannot do me any harm because it cannot grow on its own. Okay, so what does it need to do? It needs to enter a host cell. Okay, it cannot enter any host cell. It has to be specific host cell. All viruses are like that. Viruses outside of a host, of any host, is completely inactive. So there is a debate in the science world that whether we should call this living organism or should we call it dead. And so the convention is we call them a particle. When they're outside, they're a particle. When they get in, they start using our machinery and making more virus and getting out and infecting others. So that is how the virus thrives, okay? So if you don't let it go inside us, it won't be able to do anything, okay? And it does more harm, and as we go on, we'll have questions on that, and I'm sure, and we'll answer that. So inside this string-like thing that you're looking at, that dark blue string, okay, that is the genome of the virus, meaning that is those are the genes of the virus. Virus is a very light traveler. It just packs what it needs, not more than that, because it knows the host has everything else, and I get it from there. Okay, the only things that are not available in the host, that is what the virus carries. Very light traveler. The blue dots that you're looking at, those are proteins that are packing the RNA. Okay, I won't go into the details of this. I know if you have questions, I will answer that later on. So this blue, that is the lipid layer. Okay, the blue circle, sorry.
Yeah, it's touch screen, sorry.
Okay, the blue square, the blue surrounding circle that you're looking at, okay, that is your lipid layer, and through which the spike protein is protruding out. So this is how we relate the previous picture to this picture. Okay, so this virus has an RNA genome. It is made up of RNA. Usually our genes are DNA, right? But the virus can have either. Viruses are the only ones who can have RNA genome or DNA genome. Okay, and they have variety of genomes. So viruses differ from us in that way. Okay, but they cannot have both. They cannot have both DNA and RNA. They either are RNA viruses or they are DNA viruses. To give you an example, this is an RNA virus, of course. Okay, polio is an RNA virus. The common cold, rhinoviruses are RNA viruses. Adenovirus is a DNA virus. We're going to talk about adenovirus when we talk about the vaccine. Okay, herpes virus is a DNA virus. Pox is a DNA virus. Okay, so just to give you some examples. But the common theme of virus is the same. All viruses work in the same way. They get into the host and then they hijack your cells, machinery, and use it to make their own virus. And that's why we feel so sick when we have any viral infection. And you cannot do much. You'll see that there are not many antiviral drugs. Why? Because then you have to target your own cells, right? So so we already talked about how viruses are different from other germs. So to really talk about bacteria or protozoa, like for example, amoebiasis causing amoeba, or Giardia, these are single-celled cells. Okay, so they can live on their own. Okay, however, they can grow on their own if you give them food. However, a virus, no. It needs a host cell, okay? So on the left-hand side, this is how the SARS coronavirus, the COVID, enters the cell. So how? Through the spike protein. Remember I said the spike protein? So this is our cell, the pink cell underneath, this is our cell, okay? So if our cell have the receptor, which is ACE2, this is a very ubiquitous receptor. It is present in many parts of our body, unlike other receptors, okay? So if it is present in many parts of our body, many cells in many parts of our body can be infected. And we have seen that with coronavirus, right? It can infect the intestine, eyes, nervous tissue like brain, okay? Lungs, of course, okay? And it can damage other organs because this receptor is present in many places. Okay, so how does the spike protein interacts with this? So this is specific interaction, okay? Unless this ACE2 is there, the virus cannot enter. So when it binds to this, the virus is then taken in. There is a role of another protein right here on the membrane. This is called TMPRSS2. What does it do? It cuts the S protein, the spike protein, into two, and that facilitates the entry. Okay, so not only the receptor, but also that cutting enzyme, which is also present there. So these two have roles in getting the virus into our cells. So once the virus enters, then it can target the organs that are here, because once it enters, it can then make more virus in that cell wherever it enters, and that virus then spreads within your body. And any cell that has the ACE2 will take it up, okay? And then it takes up your cell, it makes more virus, and it gets out. So it not only infects the other part of your body, but it also goes to other people, okay? So here in this right-hand side, it is showing you wherever ACE is expressed, ACE2 is expressed. Okay, so the green ones are the organs where you can find ACE2, and the red one is where there are no ACE, not found yet. Okay, so how do you think you can prevent the infection? Knowing this, I ask you, how do you think, if you were on the other side, if you're fighting the virus, what would you do to prevent it from infecting us? So you know the mechanism now, that helps us, right? So you would stop, how would you stop the virus from getting in? In order to get in, the virus needs that ACE2, right? So if you can block it, that will work. So if you have a drug to block the ACE2, so they're working on that. If you can work on the TNP or SS2, that could work too. But what is a quick fix? The best quick fix would be to make a protein that will attack the S2. And if the S2 is blocked, it cannot, sorry, ACE2, to block the S. So if we have an antibody, which is a protein our immune system makes, to block the S, so it binds to the S protein on the virus, then the virus will not be able to enter because the S is blocked. There's no chance of that interacting with the receptor. Does that make sense? Yeah, so that is what we are trying to do, a fast prevention, is trying to make our immune system acquainted with the S, not the virus, just the spike protein. Just the spike protein, nothing else, okay, not the virus. Usually in previous times when we didn't have technology available, we would take the whole virus, kill it, and then give it to us. So the spike protein would be outside, and our immune system would see it and make antibodies. However, now we have technology available, so we only want our system to see the spike protein. That's it. So the virus is not getting in, nothing is going to make us sick, but our immune system will know the spike protein. And so, when the virus comes, it will attack the spike proteins and immobilize or neutralize the virus. So the virus won't be able to get into our cells. Okay. Okay, so how do we get variants? All viruses change their genome. So once they get into our cells, right, they are making their RNA, they're making more protein, and then they're getting out. So virus is more like a factory. Unlike cells, in cells things happen in a more, our cells have our own machinery. We make our own RNA, we make our own protein, and we go from there. We are not depending on anyone. The virus, okay, very quickly wants to make as much virus as possible and get out of the host. Okay, so while it is doing that, okay, specifically RNA, all viruses change their genome, okay? Why? Because they evolve. Everybody evolves, even we evolve, okay? So RNA viruses more so because they do not have an editing mechanism in their system. The DNA viruses, DNA itself has DNA enzymes have, like the DNA replicating enzymes has the ability to edit as it goes. The RNA polymerase that replicates the RNA into another RNA doesn't have that. So there are more chances of having mutations or changes in the gene of the virus. So while this happens, you get many variants. Some of them don't matter because they don't have significant changes to their characteristic, but some do matter. What do the scientists see? They want to see which variants are of concern. Why? Because they may be spreading faster, okay, or they may be causing more severe illness. So those are of concern, okay? And there may be other variants also that may not have significant difference with the original virus. So it doesn't matter. So if you look at this, there is another mutant or variant, mu, which I have not included here because CDC doesn't have any information, concrete information on that. I think they're still working on it. So we have 4 variants over here and they have named it alpha, beta, gamma, and delta. Many of you will also see the B.1, 1.7 and all those numbers. Those are hard to remember. So it is better to remember the alpha, beta, gamma, and delta. So these are different forms of the same virus, and they have, why did they form? Because we allowed them, right? Why? Because we allowed them to infect us. So the pandemic is ongoing, right? So we have people getting infected, the virus gets inside, mutates. And once in a while, we get variants which are of concern because they have changed significantly from the original virus with respect to their ability to infect, spread, or cause severe disease. Okay.
Jean Marie has a question.
It's spreading to pets, right?
Yeah, how dangerous is a spread to other species? We know it's gone into some pets.
So again, work in progress. So we know one thing that pets can be infected, they don't get severe disease. Up until now, we haven't seen any severe disease in pets. And also remember, when you're putting it in pets or it's getting into pets, it may have a chance to mutate again. So remember this jump species, so this virus came from bats, I believe, okay, jumped from species, right? So I'm not very comfortable with pets having it because you don't know what's going to happen in there. As the virus is multiplying in the pets, I don't know what it's going to do to its genome. So it is kind of an uncomfortable thought, although pets have not been seriously ill yet. So that's something we need to think about here.
And there's another question. Can we safely say we do not have the alpha around anymore? And if so, the vaccines aren't helpful as that variant is gone while a much stronger variant is here. I've also been told that vaccines were never designed to prevent the spread of the virus.
Well,
So maybe address it. What about the alpha?
Well, the alpha is gone. I don't think it's completely gone. It has become less prevalent, should you say? I don't know if the variants are going to completely go away. Okay, so there will be, the prevalence is gone, should you say. So now Delta is more prevalent. Okay, and then now we have Mu, I don't know how that is. But Delta, as you know, spreads much faster. So I don't know if that we can say for sure that alpha is completely gone. Okay, so we still have, I think, alpha, beta, gamma, and delta, but delta is more prevalent. So they are sequencing. What they do is they take the virus, when they are doing the testing, many of that goes into the labs and they actually sequence the virus. So they find out the sequence of the genome. And that actually they compare with the original virus with all these variants, because we know the sequence of the variants, and then we figure out which variant is causing the infection. And if you read, most of the cases now have Delta in there. Okay, so they are sequencing them to find out that that is Delta variant. So Delta is more prevalent, but are Gamma, Beta, and Alpha gone? Don't know. I don't think so.
Jody asked about the zoo animals. I saw that too. Some zoo animals are sick.
Yeah.
She said lions and gorillas. I saw lions and tigers for sure. I don't know about gorillas. Do you know anything more about that?
I don't, just Jody know.
Jody, do you know?
She's also a virologist.
See.
So now going to the type of vaccine. So we all agreed that if we can bind to we have something to like block the spike protein on the virus won't be able to enter us. Okay, so that's the best way to go about it. So, we want to make vaccines, right? So we have different kinds of vaccines that are available. Okay, so we all know this. Inactivated vaccines, like you take the flu virus, you kill it, and then you inject it. Okay, so then our system will get acquainted with that virus. Our immune system gets an idea of what the flu virus looks like, and we have immunity built up. So when flu virus really comes, we are able to counter that. So what is the inactivated vaccine? So that is basically what I just described. The polio shot that you get, that is also inactivated polio vaccine. What is the disadvantage? You have to get boosters because it doesn't give you a long-term immunity. So what does give long-term immunity? Something does, live attenuated virus. And I know you guys have taken this, I have taken it, oral polio vaccine. So what is oral polio vaccine? They're giving you the live virus, but it is not the original virus. It is changed to make it very weak so that it won't make us sick. But the immune system will know that it is the polio virus. We will have immunity to the real polio, the one that is really disease causing. Now, why is this good? This is good because it gives you your system, the real feeling of illness. So it gives you a long-term immunity with only one dose. It gives you a long-term immunity. However, remember, we have people with weakened immune system around us. So if you give this to people with weakened immune system, they can get ill with that. Okay, and as for example, polio, they're giving you oral polio, so it's going through the GI tract and it's getting out from the feces, getting into the water. And in many countries where they still give oral polio vaccine, they have stopped it in the United States. The weakened virus enters the water, gets into people who are immune compromised, and they have sequenced the virus to find the vaccine virus causing the infection, not the polio virus. So that's a concern. So that is why live attenuated viruses, although they're really good, they may not be good for people with weakened immune system. So we still get MMR. MMR is live attenuated vaccine, mumps, measles, and rubella. That is given as a shot, not through the way it usually travels through a system, but it is still live attenuated vaccine.
So there's another question here on vaccine. Should I ask it now?
Yes.
So the person writes, I've been told that vaccines were never designed to stop the spread. Is this true? If the vaccines were designed for alpha, how effective can they now be now that we mostly have delta?
Okay, good question. So usually the vaccines, when they're made against one virus, one variant of the same virus, they're effective against the other virus. Are they 100% effective? Probably not, but you will still get protection, partial protection from that. So for example, think about flu. You know how flu vaccines are given, right? Every year, CDC assumes which flu variant is going to come. So we don't call it variant, but we know what that means. So the CDC, from the trends from the past years, figures out a way of predicting which flu virus is going to come to North America this time. And it manufactures that vaccine, okay, and then it gives it to us, right? Sometimes it happens that that strain does not come, okay, another one comes. Do we then, what happens then? We still get protection, partial protection. You will still get ill people, but they won't be as severely ill enough. And you know how flu kills people. And so that is what I have to say about that, I guess. So you still get partial protection.
Yeah, and the same person asks, what is it about the makeup of inactivated vaccines that causes them not to impart long-term immunity?
It is said that your immune system sees it, but it does not go through the original way as it would meet a live virus or a live pathogen coming into your system. So when a live pathogen comes, it is like a real thing coming in. And when you're giving a heat killed, it is like, it's still the virus, but it's not live, or it is inactivated, should you say, because virus you cannot say live, right? So also, if you give other vaccines which are heat killed, you are giving not live. So that is I think the difference, and that's why you need boosters to remind your immune system that, hey, don't forget. So once in a while, just remind, and we may have to do this with this virus. We have to remind our system, because if your body is not encountering a particular pathogen which you have immunity to for a long time, it goes into the back burner, okay, to bring it forward. For example, whooping cough, and I didn't know this when I started working in public health. They used to send us nasopharyngeal swabs to figure out whether that had a whooping cough or pertussis in there. Okay, that's a bacteria, Bordetella pertussis, that causes whooping cough. And whooping cough is a serious illness. We all get vaccines, DTaP, that is pertussis, the P is pertussis. So when the they were sending, hospitals were sending samples, nasopharyngeal swabs. I asked, which age is this? So some of them were from babies who had not been immunized, okay, yet. But they were saying there were people 50 and above who we were getting samples from. So that is when, we came to know that we probably need a booster after 50 to, remind our immune system, hey, don't forget about pertussis. Okay, because when you get older and your immune system becomes weaker, you can get more severe illness than you would otherwise.
So can someone get the inactive vaccine and then supplement it by building their immune system?
They can, yes. It's always good to have a good immune system. So, to build a strong immune system. And immune system is also like more, so every character that we have depends on the genes and the environment, okay? So both have contributions. So what you are talking about is the contribution from the environment, right? So you're building a good immune system, but it unfortunately, or I don't know how I should put this, but genetics plays a very big role in immune system. So you inherit much of the immunity from your parents. How do they know this? They know this from research, of course, and they have found out that parents who died of an infection, okay, the kids from that parent are more likely to have more infections in their lifetime. So that is how they figured out that, and we all know that the immune system is actually more inherited. But does the nature, like nurture, not play any role in this? Of course not. You can always boost your immune system. So, both play a role, but more genetics than. So live attenuated vaccines also, all of us have taken chickenpox. This is a live attenuated vaccine. I don't know how many of you have taken smallpox. I did. I'm old enough to have had smallpox when I was born. The smallpox vaccine. I still have the scar. So that is also a live attenuated vaccine.
But they don't give smallpox anymore.
No, because it has been eradicated. And so that tells you how vaccines work. It has been eradicated because of the vaccine. Okay, so 1975, I think, or 78, that is when WHO declared that there is no smallpox anymore. So going to the subunit vaccines, we know this also. For example, Haemophilus influenza B or HIB that you get, Shingrix that we are getting now, the shingles vaccine, that is also a part of the pathogen is given, so that you are getting acquainted with a part of the pathogen, not the whole pathogen. Much like the spike protein. Okay, so if you were to just inject spike protein into me, that would also generate immunity. But much better is the vaccine that we are getting, and I'll show you how. And sometimes you get toxoid vaccines. What is toxoid? So suppose tetanus toxin, okay? It is not the organism that is, as a whole, that is giving you the illness, not Clostridium tetani, which is a bacteria, but the toxin that it secretes, okay? So what do? You get the system acquainted with the toxin, but you can't give the toxin, because if you give somebody the toxin, that person is going to get sick with tetanus, right? So what do? You change the toxin to toxoid and then inject it. So your immune system will know the toxin, but won't get sick. We won't get sick from that. So that is called the toxoid vaccine. Okay, so now coming to the ones that we have for COVID, okay, that's more interesting and more relevant to this talk. So messenger RNA vaccines are what we are getting from Pfizer and from Moderna. So what is messenger RNA? Messenger RNA is the code that makes the protein. So our genes are in our DNA, right? So our system makes, whenever we need a protein, our system gets that little code from the DNA, which is inside, and it converts it into messenger RNA. That's why it's called messenger RNA, because it gets the message only for the protein. And then it goes into the process of making the protein, and then we have the protein. So our scientists are using that to make messenger RNA. So we can make messenger RNA in vitro. So we don't have to grow the virus to make messenger RNA or get the viral genome, no. So we know the code. We have studied the code, we have sequenced the code. So all we need to do is just encode it and make it in vitro. In vitro meaning in glass, in test tubes. Okay, so we make the messenger RNA in test tubes. And then what you do, you make a batch production and then you pack it up. How? With, usually I looked at the formulation, it's all lipids, cholesterol, other lipids, okay, and you pack it up real good. Why? First of all, there are some elements introduced into the messenger RNA artificially that is not present in the virus. Why? To make it more stable, okay, and also to make your body accept it better. Also, you're packing it up better, and also for stability. So you're packing up with your lipids. Why? So it will enter your cells easily. Number one. Number two, it will be more stable and won't be degraded as it goes in. Now, coming to that, many people are of the opinion that messenger RNA is very stable. I don't know who is saying this, but messenger RNA is the most unstable molecule that is around. Why? Because if you're working with RNA in the lab and anyone comes near your bench with bare hands, you are like, get out of my lab, because it is so transient. You have to be so careful when you're working with RNA. So RNA does not, it is designed to be like that. It is a transient molecule that is made and degraded as needed in the body. And so also outside. Okay, so when messenger RNA goes into the body, okay, what it does is it makes your system cells take it up because you packed it up with lipids, right? It enters your cells. Your cell will make the protein and the mRNA will be degraded and gone. Okay, so it doesn't last very long. So you get the protein and your cells are now showing the protein. So whichever cell made the protein, got the RNA, made the protein, is now showing it on the surface. Okay, and your immune cells are seeing that and recognizing it as foreign and is building up immunity against it. So when the real virus comes in with its spike protein, your body is ready with proteins called antibodies, and they're going to attack the S protein and immobilize the virus so it cannot enter your cells. That's the whole idea. So Pfizer and Moderna have done the same thing. They have taken the whole spike protein, okay, and then they have modified it a little bit to make it more stable, to make it more acceptable in the body. There is another company who's making a messenger RNA vaccine, but it's very, very less efficient and has more side effects. Why? The only difference that I just read it before I came here, is they have not introduced those changes in the messenger RNA that makes it more stable and makes it more acceptable. Okay.
So so there's a question about the AstraZeneca vaccine. Is that the one that you were just
No.
Okay, so the question about the AstraZeneca one was that live attenuated, and so do we know, is it live or weakened or?
AstraZeneca is not live attenuated. I don't think so.
Okay.
They're not making any live attenuated coronavirus vaccines.
Oh, okay.
As far as I know.
And then also you noted that this mRNA technology has been available for decades, and I've heard this is why they could create this more quickly.
Yeah.
I'm curious what ways this was being used, or how was this being used or researched for medical use?
It was being tested for many different vaccines, but they have never used it before. So yeah, that's all I know about it. They have been testing this for decades. Whoever asked that question is absolutely right. Okay, but this is the first time we are using it, and it turns out to be very efficient. Nobody was quite sure about this. So when the results came out and we found that this is very effective, we were everybody was very thrilled, because we didn't know how this would work anyway. So what is the benefit of messenger RNA? Messenger RNA or DNA or any of those, they are the least antigenic, meaning they will not cause any immune reactions to the body. Okay, so that is one thing. Secondly, it gets degraded very quickly, and you are not putting any part of the virus in, not any part. It's just the messenger RNA, and it's going to get degraded anyway. So it's not really any part of the virus that is going in. So it is a part of the genome, obviously, but then you have changed it a little bit. So this is the only time we have ever used messenger RNA vaccines. And then we have viral vector vaccines, which are also being used in Johnson & Johnson vaccine, right? So this is where you're using adenovirus. So what do you do? Adenovirus infects your respiratory tract, okay? So what you're doing is you're taking out the gene that would cause illness, Okay, and this has been going on for a long time. Ebola virus vaccine is, or adenoviral vaccine. Not only in vaccines, we have used this for gene delivery or use in gene therapy, which is not the talk today, so I won't talk about that, but it has been used in many other ways. So what you do is you take out the E1 gene from adenovirus that is going to make the virus more dangerous. So you're taking that out and you're putting in the spike protein gene in there. So you're taking out the adenovirus gene in place the dangerous gene and you're putting in the spike protein. And then what you're doing is you're making the virus in vitro. So you're making the virus with help of a cultured cell in vitro. Then you're taking that virus, this is now a different virus, it's not an adenovirus because it doesn't have all the genes in the adenovirus, but it has only the spike protein gene of the coronavirus. So now you're giving that to people. Right? It won't make you sick from adenovirus, it won't make you sick from coronavirus, but it's going to produce immunity. So that is the other vaccine that we are using. So that is basically the 3 steps that are designed here. Similarly, once it produces the spike protein in your cells that have taken up the virus, you are then going to display it on the surface. Your cells are going to display it on the surface. Your immune system is going to recognize that, make defense against ready for that, and you are going to be okay with when the virus comes in.
So Sanhita, 2 questions, one from Jody and one from Bonnie Shaker. Jody writes, my son-in-law had a breakthrough case.
Okay.
He wanted to get a test after his quarantine to make sure he was not infectious. His doctor said he might test positive PCR for a long time.
Yeah.
I don't understand seeing that mRNA is so unstable, why this is so.
Okay, it's not the mRNA, Jody. It is the RNA itself, the viral RNA itself that is prevailing in the system. So what kind of vaccine did he get? Just curious.
Can you hear that, Jody?
I think he got Moderna.
Okay, that is highly unusual. All the breakthrough cases that I'm hearing are Pfizer. So this is something different, but anyway, breakthrough cases can happen and I'll tell you why. Did he get any illness? Like he probably-
Not much, a couple. My daughter-in-law said no, I guess he posted on Facebook, he had some symptoms, but not much.
Yeah, so I'm going to talk about the breakthrough right now because Jody brought it up.
Okay.
So when breakthrough infections happen, okay, so what are we doing here? We are giving, so your son-in-law already, whoever has been vaccinated already has the antibody. But these antibodies are inside, okay? The infection is happening through your mucous surface, meaning inside, like when you're inhaling the tract that goes, respiratory tract that goes in, okay? These antibodies that are formed are not designed to work like that. It is like, let me give you an example for everybody. So suppose you're guarding this door, you're guarding the door, right? So nobody can get in, right? So there is another way of protecting this house. How? You're not guarding the door, so the intruder thinks that it's okay to get in, right? But inside you're armed. So as soon as the intruder enters, you are jumping into that intruder, right? So what we are getting today, the vaccines that we are getting, the army is inside us, but not on the surface, the mucous surface. So when breakthrough cases are happening, because there is no antibody, now Jody will know there is no IgA to induce IgA, you have to give the flu mist vaccine, that generates IgA. That generates protection on the mucus, but it's not very effective, so they've stopped giving that flu mist. Remember they used to give a nasal spray? Okay. Also, banana vaccine for diarrheal diseases. So they make you eat a banana with the encoded DNA and it generates IgA, like the antibody on the surface. Okay, so these antibodies are inside. So breakthrough cases are happening because the virus is getting and multiplying in the mucous surface. But then as soon as it gets inside, we have antibodies already there, right? So inside, the army is waiting and you jump. And so the preliminary symptoms are not even, okay? Sometimes you don't even get any infections or very mild infections. By then the other antibodies come into action and they are protecting. So that is the breakthrough case. We don't have a vaccine on the surface, that will act on the surface. And I don't think it will be very effective because we tested with flu, which is a respiratory virus, and that didn't work. The second question.
So this is from Bonnie Shaker. She says she has a question about scientific practices intersection with public health and education. Should or how can scientists educate the public on its practices? For instance, the form Pfizer had to fill out for human trial approval was the old form for live or attenuated virus vaccines. So included language about shedding and being near pregnant women, which isn't relevant to mRNA technology. But it is that form that was used to spread misinformation about COVID-19 vaccine and fertility. So again, what responsibility does science have and how can it educate the public on its practices?
I'm totally with you, Bonnie. There's so much going out there that is not relevant, and many people are thinking that, I just showed you a picture where there is they're showing you that ACE2 is present in the ovaries, and so people are like, are we going to get sterile because of if we get an infection from this virus? So I know many questions are unanswered, but if you know something is wrong, you should not be circulating that. I'm totally with you. So scientists have responsibility too, to prevent this from happening, to get information that is wrong outside. So we as scientists also, and those specifically, those who are working in the lab are really, really responsible to not let this happen. And that is why we are standing here today, because of so much of bad information that is going out that is preventing people from getting the right kind of information and going forward with that. So people are stalling right there and thinking, well, am I doing the right thing or am I not doing the right thing? And I don't blame them.
So where would you suggest to people to get information? Like if you really want to learn, where do you even start?
I would wait, and I would read the FDA inserts. If you're really, really interested, go to the FDA or go to university sites. Like, for example, Stanford has information, Yale has information on their website, medical schools have information on their site where doctors or scientists who are actually working on that are giving you information. And remember, the information is changing. So I know what Bonnie just said, that is unexcusable, because you shouldn't write something or use a form that is not valid. Specifically for this important of a situation. But I would I think, yeah, you have to go to sources which you can actually trust. I know even CDC was giving wrong information, and not wrong information, what they believed was right at that point. And then they changed as it goes. And every day, WHO, CDC website is changing. So, in order to keep updated, I think you have to go through that every day almost to see if anything is changing.
Now, Jean Marie asks, how dangerous is it for the continuing spread of COVID-19 that countries worldwide are lagging in vaccinations due to lack of vaccine?
Jean Marie, you know better than I that this is allowing more variants to form. And you know that if you allow more variants to form, you know what happens, right? So one of them may be more dangerous. We haven't still encountered a more dangerous virus than the original. So I think Alpha was a little bit more dangerous, but Delta, although it is spreading more fast, it is not more severe. It's not causing more severe disease than the original. But we may not be lucky to do that. So the countries that are lagging behind in vaccine, okay, I'm really worried about it. Very worried about it. So they can prevent the spread though if they are not vaccinated by wearing masks. So there has been a study from Yale and Stanford on wearing masks and how it is effective. So they have done the study in Bangladesh. And it is not possible to socially distance in Bangladesh because there are so many people in such a small area. Okay, and they have found that wearing masks is really effective, and specifically wearing surgical or medical masks are more effective than cloth masks. So that is their conclusion from their study, and they're still doing more studies on that.
Okay, another question here. Our cells recognize virus after a vaccination. How is it that our cells, after a few months, forget what the virus looks like? Why does not no longer recognize the intruder?
Well, I don't think it completely not recognize the intruder. but its efficiency falls. So it is like, suppose your army, is not going out to fight anymore and is not practicing anything anymore. They're just sitting there, getting up late in the morning, drinking coffee and doing nothing. And something happens. A war comes. Do you think your army is going to be able to work right away? No, right? So that is why you need to train your immune system once in a while. So that is why the booster. Okay, the immune system is designed to not work if you don't encounter the virus. So slowly it goes, it is still there, but it's not active anymore. So in order to bring it to the forefront, you have to remind it, hey, don't forget coronavirus, it's still around.
Yeah, isn't there something I also read about the time between the first, in the case of Pfizer Moderna, the time between the first and the second shot was 3 weeks or something, and normally it would be a minimum of say 6 months. And so because it was compacted into the shorter time frame, that's another reason why we may need a booster.
Yeah, that, and also remember, we do need boosters for many vaccines. So all of you guys take tetanus every 10 years, right? Why do you do that? Why take tetanus every 10 years? Because you're boosting your system, right? That's a toxoid vaccine. That's not even an mRNA vaccine. And you are more likely to encounter tetanus than any other thing. That's why you take that shot. So we take boosters all the time,
Someone else asked here, do we know where the virus came from, and could that information help us with fighting it?
Well, according to publications, it came from bat. I know people have been saying that it is an accident from a laboratory. Nobody knows. There has been investigations, and we still have not been able to conclude anything from there. As a scientist, I believe that will not help us with the present pandemic anymore, because it's already out and people are already getting sick. So we need to get rid of this pandemic before we can even think of anything else and how to prevent that from happening in the future. Okay, so people who work with recombinant viruses or viruses themselves are pretty careful in containing it into the lab. So I hope it won't happen again. If it did happen this time, I don't think it did, because there is no evidence to suggest that it happened.
So someone else says, why do some people have more harsher side effects from the vaccine than others? Is it because they have a weaker immune system, or?
I don't know whether they have a weaker or a stronger immune system, because obviously the side effects are because your immune system is working. Okay, so think about it. You're given the shot, then about 24 hours later, for mRNA vaccine, it will be more time will be needed, because protein has to be made, and then the immune system will see. And the immune cells actually, when they react, they secrete some substances that can cause fever and other things. Very much, because whenever immune system works, that is when you get sick, right? That is why when you have a viral infection, the immune system works, and that's why the sickness, right? So that is a part of it.
Okay, why is it that vaccinated and unvaccinated people have the same viral load?
Okay, the virus may be multiplying in the nose area or the mucus area, but it may not be causing any problem to you. No, and another thing which is I don't understand is there are many people, 40% or more people are asymptomatic even when they get infected. And maybe their immune system is super good. I don't know. Last pandemic, when there was flu pandemic 100 years ago, the people with stronger immune system were more sick than people with weaker immune system. This time it appears it's the opposite. It's the weaker people, weaker immune system people who are having problem, or people who have comorbidities. But last time when the flu pandemic happened, it was the opposite. If you read the year of the flu, you will see young people with stronger immune system had it.
Okay, a couple of questions. Jody asked, what do you know about the cause of long COVID? Long COVID?
I don't know, Jody. But we don't know what the long-term effects are anyway, because I think the study is ongoing. I don't know if Jody knows something about it.
Jody, do you want to comment?
I don't really know the cause, but I mean, it's the fog, the brain fog that can go on for months, and lack of smell, and people that are really tired for just months and months and months.
Jody,
I heard they didn't know why it was happening.
yeah.
Just wondered if you'd read anything currently.
The problem is that there are receptors in all the sensory organs and the brain.
I know.
It's a problem with this particular virus, because had it only had receptors in the lung, then we would have been much better with this. And even blood vessels, that's the problem. You know, if you have receptors, they are going to get affected. So you see the little kids who are having Kawasaki, not all of them are having, thank goodness, but you know, people are having Kawasaki syndrome. Why? Because of the blood vessels. So, and then also eyes. It's amazing.
So somebody asked, she says, I had bad side effects from the vaccine. Will I experience that again when I get the booster?
You may or you may not.
Don't know. Okay. I'm reading that the FDA has not verified the need for the booster shot by Pfizer
Yeah.
because the data isn't verified. Is that booster shots aren't really that necessary or helpful, or?
Well, this debate has been ongoing for a long time, for many months. Many people are saying, well, we have gotten 2 mRNA vaccines. We don't know, at least we know something about the adenovirus delivery system. We don't know anything about the mRNA. Has it changed anything in our system? Anything at all? They're not saying DNA or anything. Anything at all. We don't know that. We won't know that for many years to come. We haven't seen any change yet. So hopefully there has not been any change, because that messenger RNA, if we know the theory, has already gone out of our system already. So it should not cause any genetic difference, but does it have any other change? I don't know.
It's safer to take a traditionally made vaccine?
It's going to take a long time to make a traditionally safe like vaccine. Maybe later on they will come up with that, because you know, but these vaccines have been so effective that people are not thinking of making any traditional vaccine. So I know in India they were giving a heat kill vaccine in India. They're calling it Covaxin or Covishield. I don't know which one. So
What about AstraZeneca?
yeah, that is inactivated. It's not been given in I thought you were asking about the United States. In India, they're giving traditional vaccine, I know.
Someone's asking about Sinopharm?
No, I haven't.
You don't know that?
No, I don't know about Sinopharm. I think I read it, but I don't remember like much about it. Is that Sinopharm is the one who have 47% efficiency? Vaccine, messenger RNA? I don't know. I don't remember the name of the company. So basically, I think all of you know about this, so I won't go into this. So that Professor Meckenbauer actually asked me to show you this. So vaccine efficacy for all the vaccine that we usually take are on this slide. And then the COVID vaccines are on this slide. So this is pretty efficient. These vaccines are very, very efficient in preventing serious illness. And that's what it is supposed to do. If it prevents a spread, like there was a question before, that would be great. We don't know. People are saying that people who have been vaccinated can spread the virus. In the very beginning, they were saying it cannot. And now they're saying they can. So
Because it would be that mucus area.
right. So it would be still they would be able to spread. So they are advising them to actually, if anyone who's vaccinated gets positive test, to just seclude themselves for a while.
Yeah, what about the-
Yeah, Johnson and Johnson, really, I agree with you. There have been many blood clot concerns. And I was reading a paper on that was discussing the mechanism why of that. So I don't know, although, because we have used adenovirus before for other purposes, as I said, and on sick people. So this is on healthy people. So I thought it should not give you that problem. Kevin, you had a question?
I was just wondering your thoughts on, like, you know, will it ever be eradicated, or is this just
It
something like the flu, like that we're gonna have to deal with, which we have for hundreds of years?
So good thing you asked, because it is an RNA virus and a respiratory virus, it's going to be hard to get rid of it, okay? For pox, smallpox has been eradicated, right? It's a DNA virus, first of all, and it only had humans as a host. Now we are hearing that pets and animals are also hosts for this coronavirus.
Which makes it
Similarly
even
for
more
influenza
difficult.
A, so it's going to be harder to eradicate that.
So there's been some new studies in Israel that suggest that natural immunity from recovering from COVID is just as effective as vaccinated immunity. Is that true, or why or why not?
So it could be true, because I would think for all other diseases, they always say that natural immunity is much better than vaccine-induced immunity. I had heard some confusing remarks in the very beginning saying that here, in this case, the antibodies are going away in like 3 months or so from the natural immunity. So when you have the disease, the antibody titer in blood is falling. So that doesn't mean though, later on I talked to some scientists and they told me that the immune cells are still there, the ones that have been encountering the virus. So they should not cause a problem. So although the antibody titer is going down, which happens, it wanes with time, but then remember your immune cells are still here to function. And so all you have to do is remind them. So I think with the natural immunity too, it will wane, but remember, they still have all the immune cells geared up there. So that's what they answered, the scientists who were working on that.
And what are your thoughts about the use of alternative medications that can either treat or prevent COVID? There's been talk about like ivermectin, hydroxychloroquine.
Okay, so chloroquine is used to treat parasite infection, like malaria. Ivermectin similarly is used to treat parasite infection and worm infection.
Yeah.
So I don't know how they're going to prevent viral infection, because if you're going to use a drug for antiviral purposes, up until now, from my training and all I've read and the literature, you are going to inhibit one of the processes of the virus. So like maybe getting in, like for example, Tamiflu is given for preventing flu from getting in. So nurses who work on the flu floors are not only given the vaccine, but also Tamiflu to prevent it from, it's like a prophylactic drug that prevents. Okay, malaria, similarly, you use chloroquine, which they are saying, well, that is, it works in a different mechanism. I don't know if those drugs, ivermectin or quinine, can prevent any of the viral processes that are happening in the cell. So if they can, I don't know if there is any evidence of that happening in vitro at least. We will get an idea of what's happening with those drugs.
And so what are your thoughts about supplements like vitamin D, zinc?
It's always good to supplement your immune system, always good to boost your immune system. So keep taking the vitamins, I would say, and keep taking zinc and whatever you want to, because that, will only help you. So.
Are we now in an endemic, or is an end in sight for this?
I hope so. Now, we are going to end this pandemic one way or the other. My question is, which way is this? Are we going to kill more people and end the pandemic, or are we going to take the vaccine or everyone, get protected and we are going to end the pandemic? I don't think, like Kevin said, it's probably going to continue for many years, like the flu, because it's a respiratory tract virus. And as I said, there are other animals getting, like flu A, influenza A. Influenza has 3 viruses, influenza A, B, and C. A can infect humans as well as other animals. B and C can only infect humans. So we are not so worried about B, and we are worried, not so worried. But A is something that can jump from here, and then they change, they mutate, and then they make mix and match, and then you have a new virus. I hope Corona doesn't go there. That's all I hope. But for influenza, it's a different virus and it has a different RNA get up. So I won't go there, but it has it. So Corona probably won't do that.
Well, we're just about out of time here. You want to have some concluding remarks, or I know?
No, thank you so much, all of you, for being here. I also learned a lot from your questions and also from your insights into this. Because I can see all of you have read also as much as I have. So, thank you all for coming and
Well,
being here.
well, we thank you. So this Q&A came about because I was asking Dr. Gupta so many questions over the summer, and she's so incredibly knowledgeable about the subject. I said, I really think we need to have a Q&A. So we're really grateful for your expertise.
It's all
and